Hello everyone, welcome to the third lecture of our short course. In today's video we will discuss theoretical underpinnings and analysis strategies in GWAS. We will explore the use of linear regression in GWAS and discuss why it falls short, why it doesn't work. And then I will introduce linear mixed models in GWAS setting and explain how they address the issues inherent in linear regression and the further challenges they present. So let's get started. Now imagine you are interested in a question whether a particular position in genome is associated with a trait of interest. And because human genome is diploid, you have two allele copies in that position, and let's say they just have A and T alleles in that position. Then you have three possible cases. You might have both alleles have T nucleotide, or one allele is T, the other is A in any order, or both alleles are A. Then you can run a simple linear regression as follows, where Y is the trait value or vector of interest, and G is the genotype values, which can be recorded as a number of A alleles that an individual is carrying. For example, if an individual has TT genotype in that position, then this G value is 0. If that genotype is AT, if that individual has AT genotype in that position in their genome, then it has the G value 1, and so on. And beta is the slope that we are interested in, the effect size of G, and epsilon is the random noise, which is, we assume, that normally distributed with some variance. And we might get some kind of a box plot like this, where we illustrate the differences between individuals' phenotype values that carry different genotype values, TT, TA, and AA, and we might see even this kind of a slope. And we might say that, okay, carrying an A allele might be associated with having a higher phenotype value because of this slope. However, there are some defects in this kind of analysis, and basically there are some confounders that's causing that the linear regression setting does not work in GWAS analysis. And here I present the three most common confounders that are known in GWAS setting. Number one is population structure, which is the effect of two different or more different populations on our analysis setting. So for example, if you're interested in disease, where you have cases and controls, and you sampled cases only from one population and controls from another population, then any differences between the populations would be attributed to the differences between cases and controls, which could lead to false conclusions. So this is therefore a confounder, population structure, in this linear regression setting. The second confounder is related individuals. In a standard linear regression setting, we assume that the error terms are IID, or it means independent and identically distributed. In order for this to happen, we must have kind of a random sampling, but in a GWAS setting, we have so many related individuals. So for example, here I present you an example that John Krasinski is quite tall, which is an actor starring in The Office in the US version. But his brothers are even taller than him. So it is some kind of a genetic flowing on their family ancestry. And if you sample individuals that are related, then these error terms become related as well. So it breaks down the assumptions, the fundamental assumptions of linear regression. And the third confounder is the linkage disequilibrium, which is causing false conclusions in the linear regression setting, in the sense that when we are making an association analysis, if we found an association, it could be either a direct association of a variant with a trait, or even if it is not associated with the trait, there could be an indirect association due to the linkage disequilibrium between genetic variants, and therefore we would get false conclusions due to this linkage disequilibrium. So this is another confounder. So because of these known or also other possibly unknown confounders, linear regression setting is not working. And now in the next slide, I will explain how we can define a linear mixed model setting. But before that, I would like to talk about why we are not considering multivariate linear regression setting. It's not suitable because one, there is a multicollinearity in our dataset, which means that there is a high correlation among predictor variables due to the strong correlation between the genetic variants. We cannot really run multivariate linear regression. Actually, this can be fixed by using penalty terms and some kind of other methods. But the second problem is even more important in the GWAS setting, which is the low statistical power. Because of the high degree of freedom due to the large number of predictors, you have very low statistical power to detect anything, which is the fundamental argument in the GWAS. We are trying to detect associations between genetic variants and there's diseases or traits of interest, and when we run a multivariate linear regression with so many variants, we would have a very high degree of freedom, which would cause us very low statistical power. There could be also another reason about the computational challenges. I am not familiar with that, but the main two reasons, or maybe the main reason, is the low statistical power, and there is also a multicollinearity issue as well. So that's why multivariate linear regression is not suitable. And so that brings us a challenge that we cannot run a linear regression setting in GWAS. So what we use is typically a linear mixed model setting, and there are different ways to apply the linear mixed model setting. So here the one that I present is from Jiang Zhang Qi et al. Paper, which is a very good paper explaining in detail about the methodology of the GWAS in linear mixed model setting. Basically, it suggests that we have a phenotype and you have something called fixed effects and random effects. And that's why it's called a mixed model. So we have G times beta. G is the variant to be tested and beta is its effect size. So this is the thing that we are going to estimate. And we have a matrix of covariates, which I will explain in the next slide. These are fixed effects that correct for the confounding structure that I was presenting in the previous slides. And alpha C are their effects. And G is the total genetic effects, which is distributed as a normal distribution with around 0, with variance pi times sigma G square, where pi is the genetic relationship matrix. And so this only contributes to the variance of the trait, and E is the residuals of this model. And in this model, I will talk about in the next slides how this model solves our issues that we outlined for the linear regression. But before that, I would like to talk about how the association analysis framework work from this linear mixed model setting. So what we does is we looked into the generalized least square solution of beta, which can be written like this, and its corresponding variance vector of the beta hat can be written like this. And if you look at these carefully, the only unknown variables are sigma G square and sigma E square, where pi is the genetic relationship matrix, which can be computed. So these variances are the only variances, the only unknowns in this in these equations. They are usually estimated through something called RML methods, restricted maximum likelihood methods. But after that, once you get your beta hat and its variance, what we do is we define a test statistics and then apply a hypothesis testing, assuming that this follows a chi-square distribution with degree of freedom 1, and that is our null hypothesis, and then this would generate us a p-value which would then tell us about how we would be confident about concluding that the variant is associated with the trait of interest. And we would also have an effect size. If we are confident that that is associated with trait, we would also have a measure of how much it is impacting the trait. So this is the linear mixed model setting. Now I would like to talk about how this linear mixed model setting fixed for the confounding factors. Now let's start with, first of all, the population structure. Now in 2008, Novembre, Johnson, and Bragg et al. Presented an amazing paper which called Genes Mirror Geography within Europe. And what they did was they looked into the top two principal components of the genotype matrix. And what they found is that in that genotype matrix, the top two components actually mirror the geography of the Europe map. So if you look into this map carefully, you will see that it really resembles the geography of the Europe map. And by adding the top PCA components, this would allow us to control for the population structure. So if I add the top 10 or top 20 PCA components, that would definitely correct for the population structure. Indeed, it is guaranteed by this paper that the top components contains the population information, and by adding these here, we are allowing them to have separate means in those different populations, which would allow us to correct for that confounder. There are also other kind of common components that we add here as correcting for the confounders. These are age, sex, or batch centers, because these variables also might differ between different groups. For example, males and females might have different average height in different populations, and therefore you might want to correct that as well. Okay, so that being said, the second confounder was family relatedness, and linear mixed models also control for that. How they do that? So the total genetic effect G has here is a matrix called pi, which is the genetic relationship matrix. If we only use unrelated individuals in our data set, the unrelated individuals are usually defined as they are genetically distant from at least fourth degree relatives. And if you only consider unrelated individuals, then this matrix would be identity matrix. And so whenever this is identity matrix, this G plus E can be considered as like a residual itself, and so this would reduce to linear regression. So this setup is actually an extension of linear regression. And what it allows us here to adding pi in the genetic component is that genetically similar individuals will have similar environmental or residual contributions because of this G component, which contribute to the variance of phenotype. If two individuals are genetically similar, then this pi would contribute the same variance component for those individuals in their phenotype. Of course, there's also an additional residual component, but this G component, which has an effect on the variance of the Y, is influenced by this pi vector, and therefore genetically similar individuals have similar environmental variance contributions, which was not the case for linear regression setup. And by adding pi, this controls for the family relatedness. Okay, the next confounder was LD. And for that, what we do is in that setup, we apply something called leave one chromosome out, or LOCO short approach. And what is that is simply if a variant is tested in chromosome 1, then we use pi the genetic relationship matrix, which is calculated by only the variance from chromosome 2 to 22. So we exclude chromosome 1. If a genetic variant that's going to be tested is in chromosome I, then I use all autosomal chromosomes except chromosome I. And that would lead us 22 many pi genetic relationship matrices. And for each association testing, we will be using only one of them. That is something computational tractable. So instead of calculating one genetic relationship matrix, we are calculating 22 genetic relationship matrices, which is computationally tractable. And why this works, or why we are using this LOCO approach, I will explain in the next slide. So first of all, LOCO removes LD effects of markers from the same chromosome, right? So in the same chromosome, you have most of the time that includes the high LD chromosomes with the chromosome that with the variant that is being tested. And the reason for that is that LD structure is getting weaker as the distance between the genetic variants increases. And because of that, the LD effect that removes the confounding factor from the same chromosome is important. It is not the ideal setup in terms of statistically, because we would only want to exclude the high LD variants that is linked to the variant being tested. However, that option is computationally intractable, because that would lead us at least tens of thousands genetic relationship matrices, which would be computationally impossible to do even for one single GWAS. And therefore, LOCO is giving us a computational tractable approach. Well, you might ask why we are using LOCO at all, why we don't use all chromosomes at once. That would give us only one genetic relationship matrices, so it would be even computationally more tractable. Why we are using this? And the reason is, going back to this paper in 2012, Litzgarden, Lippert, and Caddy et al. Showed that they compared the case where they use all chromosomes in a mixed linear model setting, which they called it MLMI. So it is all chromosomes are included. And the second approach was MLME, which is the LOCO approach. And they showed us that if they use all chromosomes, that would reduce the power, and they define a term for that, which is proximal contamination, and which is basically saying that when you use all chromosomes to calculate your GRM, you are also using the variant that you want to test for association. So you are using that variant twice in your model, which is why they defined this term contamination. And that is the reason why LOCO is the standard gold standard approach for linear mixed model settings. And that's why it is kind of a balance between fixing this confounder in a computational tractable setting. Now, I would like to finish this with further challenges. One further challenge is what we call multiple testing burden. Imagine you are solving 100 questions and you are doing 5 mistakes. If you project that for about solving 1 million questions, that would make you have 20,000 mistakes. That's a lot of mistakes. So in order to reduce this number of mistakes, you need to be really, really careful about your judgments in these questions. And when we are running GWAS, we are basically running a hypothesis testing for each genotype variant. And there are so much genotype variant to be tested. This would allow us to have so many mistakes. And that is a kind of a burden that's still an ongoing. And for that, usually what happens is that you choose very stringent thresholds, which are usually called GWAS thresholds, that would make you have very little mistakes, even you have so much multiple testing burdens. Or there are other ways of going around of this. There are other methods that I will not going to discuss. But the logic is that there is this kind of issue that if you try to solve so many hypothesis questions, then there is a possibility that you might do so many mistakes. And in order to prevent this, you need to be really confident to make a conclusion about a hypothesis question. And the second problem is, which is kind of related with multiple testing burden, because in a multiple testing burden you want to be confident, and for that you need statistical power, and for that you need large scale data sets. And indeed GWAS requires large scale data sets because in order to be able to find some associations, we have seen in the literature in past decade or so that GWAS needs at least a amount of 100,000 individuals to find some kind of substantial associations with the diseases. Here in paper, figure taken from a paper from Tam, Patel and Turcotte et al., they looked into 3 traits: BMI, height and weight. Excuse me. And they looked into sample size on the x-axis and the number of loci they detect in the GWAS analysis. And as you can see, this as sample size is about 100,000, there aren't much associations found. Only then, when the sample size increases significantly, they were able to find significant associations. By the way, I apologize for making a mistake about the phenotype values here. It is BMI, height, and weight adjusted BMI. Okay, lastly, so we said the GWAS requires large-scale data sets, and so another challenge is that obviously a GWAS software that's capable of handling large-scale data sets. So these are the still kind of an ongoing challenges in GWAS. And that being said, this brings us to the end of today's video. We've covered the theory behind GWAS as well as why linear mixed models are the standard approach in GWAS. And in the next video, I will be talking about meta-analysis and why it matters in the concept of GWAS and how does that affect the challenges, how does that solve or mitigate the challenges that was present in linear mixed model GWAS settings or GWAS settings at all. So thank you for watching, and I hope you enjoyed the lecture, and I will see you in the next video. Bye.